Okay, so translation. And we started talking about this the other day, I remember. But translation was the event of reading your mRNA. And where are you doing this at? Perfect. So, the reading of mRNA. The ribosome. Just because it's a great question, some facts about ribosomes that you should remember. One of the first facts would be that a ribosome is made of 2 units. One is a large and the other one's a small subunit. And actually those pieces come together when the mRNA shows up. There's a small piece and a big piece. And those small and large subunits are actually made of 2 things. They're made of rRNA, which was made where? Do you remember? Where's rRNA formed? It's made in the nucleolus. Back in the day. Nucleolus is the site of rRNA, ribosomal RNA synthesis. And it's also made of associated protein. So rRNA plus protein, where rRNA is made in the nucleolus, is the constituents, the stuff that makes up ribosomes. And then not only is translation occurring at that ribosome through the reading of mRNA, but it's going to utilize the action, or the carrying potential, we could say, of tRNA molecules. And these molecules carry your amino acids. And we had gone over this drawing the other day that kind of covered the structure of these two in tandem. To review that concept, let's draw just a little piece of mRNA. And let's point to a specific codon on that mRNA. Let's point to a codon that was reading AUG. So this right here is a codon. Codons are always on the mRNA. Remember, this mRNA was derived from DNA through transcription, possibly processed if this was a eukaryotic cell. And meeting up with that codon, AUG should be an anticodon on a tRNA molecule. And if we remember, our tRNA molecule was like this very elaborate structure, but we're not going to be so elaborate. The anticodon on your tRNA would be what? Perfect. I wouldn't put a T, like as in TAC, because tRNA only has U's and not thymines. Correct? And what is this tRNA carrying, aside from the fact that it has an anticodon? What should be connected to that? And does anyone remember the name of this amino acid? Close. Methionine? Methionine. Very good. So the anticodon UAC is carrying the methionine amino acid to the AUG codon. Remember, the AUG codon was always the start codon. It signifies that this is where we are starting the process of doing translation. So, to end that little section, want to remember that mRNA has codons on it. The tRNA has the anticodon. Good, good? Okay, so let's also remind ourselves about tRNA molecules, that not every tRNA molecule is the same. They're different. And their purpose is to take amino acids to the ribosome. So they will take amino acids to the ribosome. And we should recognize, since they are all different, since there's different tRNA molecules, the amino acid that they're going to take is based on their anticodon. So the tRNA molecule is going to carry a specific amino acid, not just any given one, and that's based on the anticodon that it contains. So this tRNA that I've drawn up here, its shape with that UAC anticodon could only ever carry the methionine, and only that.
So there are just like a lot of like tRNAs floating around.
And they're not all identical. It's like saying that there's these little units of movement, these tRNA molecules that look very similar but are different at a very, very like chemical level, and their differences are what allowing them to carry different amino acids.
Would there be like 20 because you said there's 20 different amino
There would actually be 64 because there would be
acids?
64 different
but then
codons.
they're just for-
Because of that fact that there were 64 different potentials of codons. So yeah. And it's actually a little bit less, but it's pretty close to 64.
Because it starts and stops.
Yeah.
Does it work?
Start and stop.
Well, and usually the stop ones aren't coding for an amino acid.
Yeah, so
And
that's what it would be more.
now the problem that we would have is that after a period of time, the amino acids, of course, once they're taken to the ribosome, your tRNA molecule would be blank. So we have to add new amino acids. We have to add them. We need to place them on. Would need to be placed on an empty tRNA molecule. Okay. So once this tRNA molecule takes its amino acid to the ribosome, where it's reading the mRNA, it would leave off blank, and it's got to become recharged. It's got to become, it's got to start carrying a new amino acid. Now, where do you think it's going to get that new amino acid from? Pretty simple logic, actually.
Do you-
From your diet. You don't eat protein just to make muscle. You eat protein so that you can break it down into amino acids so that you can make new proteins on a cellular level. Not muscle, just enzymes. Cool. So we can imagine a case where we're starting out in the before, having this tRNA molecule that's empty, no amino acid on it, and we need to end up with a tRNA with its specific little anticodon right there, having an amino acid added to it, right? This reaction is going to require a specific enzyme, and the name of the enzyme that's required for this process is called amino acyl tRNA synthetase.
Let's have an abbreviation for that.
There is no abbreviation, I don't believe. Maybe. Amino acyl tRNA synthetase. As well as ATP. So an enzyme is helping to facilitate the rebuilding of your tRNA molecule, or the recharging, we could say, of your tRNA molecule. And if we look at the little picture right through here, you can see that you're starting out with a tRNA that's blank, correct? And it's going to be recharged, which they call this the charged form, with your new amino acid, by this enzyme, which is the amino acyl tRNA synthetase enzyme. And you'll notice that that will use some ATP along the way to get to its energetic state. So now you're starting to see the implications of cell respiration, because you know that that ATP that we mentioned there isn't free, and you know that it's formed by cell respiration. So you're not using cell respiration and the production of ATP just to move your muscles and help you think. You're using it just so that you can make insulin because you ate lunch, or hemoglobin to carry around your oxygen. You need ATP on not just a physical mirror looking at your muscles move level, it's a cellular level that you would need this energy. Good? The other day we mentioned, and this is to kind of change gears a little bit, but we mentioned something called the wobble effect. Thank you, guys. Thank you. No, it's not, but I could put it there maybe. This was an effect, this was an occurrence where there was a flexible pairing. At the third position. And a codon. If you remember from that codon chart, and we looked at it the other day a little bit, let me show you a good example of this wobble effect. Let's say that you had an mRNA sequence, and I'm going to give you 4 different codons of mRNA. Let's say that one sequence was CUU, and one was CUC, CUA, and CUG. I'll let you write all those down real quick. Let's say that those are all some sequences, a codon of mRNA. So all are a codon of mRNA. But that's not the only similarity between these three. The other similarity is that they all code for leucine, which of course is one of your amino acids. They'll code for leucine. Hopefully you can see what the difference between these 4 are. They've all got the CU in front, but you'll notice that the last position could be a UC, A, or G. How many different possible options are there in that 4th position? 4. There's only 4 bases, so you can only have 4 different deviations. And see, you can have them all. I could have put anything in that 3rd position, and I'd still code for leucine. Does that make sense? So this wobble effect, it's a flexible pairing at the 3rd position, and that's true, but it also can be a way in which mutations don't matter. So it can cause DNA mutations. To be irrelevant. If you mutate the DNA that would code for the mRNA in that 3rd spot, doesn't really matter. This would make that would make those DNA mutations silent mutations because they wouldn't change the system at all. It's like if you cut down, or there's that saying that says, like, if a tree falls and no one's around, does it make a noise? It's this kind of the same concept a little bit, because if it doesn't cause a new amino acid, then it doesn't change the protein structure or function at all. So it really isn't a mutation, you could say. It doesn't change anything, right? Okay. Now this doesn't work every time, but it works a lot of times. Okay. Let's look at something different. And let's look at our ribosomes. Remember, the ribosomes are made of tRNA, or RNA, I apologize, and they're also made of protein. The rRNA was made, of course, in the nucleolus.